Welcome everyone to our team project pitch presentation. This is very exciting. It's the culmination of our Hack 4NF event that we started on October 2, and today, November 13, we have a very exciting line up of projects that solved or trying to solve the track and problems that we submitted to these amazing teams. So during today, we will record this webinar and it will be available on ctf.org in a few days. And you have closed captions available. As I said before, click on the CC button on the Zoom bar, and you will the titles will appear. So the rules for today, what's going to happen? It's very important. So we will play all of the 3 minutes project presentation recordings on a rolling basis, and we will pause for 1 minute after each presentation so that Lucas will be able to load up the next video, but also allowing questions to be submitted in the chat. So we will not have all of the themes available to speak, but you can write the questions in the chat, and the project members for that specific project will be able to answer the questions. So if someone doesn't reply to your question or answer your question, is because they're not here present, or they don't know really how to address it. So it's really in their hands to answer questions, but we would encourage you to submit those questions anyway and start a conversation about the submissions. What will happen after this event of today? Of course, the final deadline for submission is today, 11:59. So you still have about 12 hours to work on your project. Today's pitch is not part of the competition. Today's pitch is, celebrating your work. So we know that we have some presentations that are like 20 seconds over the 3 minutes. We are not going to cut them. We're going to play them because the pitch is part of the submissions, but it's not the critical part. The judges will look at your entire projects. It's not because you're just overdue for 20 seconds. And we want to, listen to all of your projects, of course. Next week, starting tomorrow, all projects will be reviewed by all our amazing judges. And on Tuesday, November 24 at 11, we'll have another webinar and we'll announce the winners. So we will make sure that we circulate an invite for that webinar so that you can register and listen. Another note for November 24, we are also organizing a panel discussion with some experts, with mentors, with the patients, with the participants, and with a judge to understand how did this hackathon came around and what are the impressions, how can we further improve on this type of events. So why are we today? And the most important part is, of course, this is the prize pool and submission day. So as you know, we have 6 prizes. 3 top teams will win $7,500 each. So we're gonna have 3 winning teams and 3 incubation prizes. 1 of 7,500 and 2 of 5K. And what is important is to know that we are not awarding each single track, but we will award the best 3 teams overall as the winning teams. And then you can still submit an incubation prize in submissions by reading the submission criteria that were clearly stated on the rules. So in order for you to get an incubation prize, you need to submit a 3-month plan, 2-page max, that includes different sections like brief project summary, impact of your solution, and a plan for the development of the projects, including clear goals. So this is the document that will be reviewed for the incubation plan. So go back and read those official rules. If you haven't clearly submitted that document, you still have 12 hours to do so and compete for a grant. And this event would not happen without our amazing sponsors. So I want to introduce our two presenting sponsors, Salesforce and GraphPad. Thank you so much for your amazing support. You really made a difference, but also our general sponsors and data partners, starting from NTAP, the Neurofibromatosis Therapeutic Acceleration Program at Johns Hopkins, the Gilbert Family Foundation, and then Bougie, Aptec, Helix, Sidon, Sage Bionetwork, Arcturus, Elsevier, Genuity Science, Sarexis, AstraZeneca, DNA Nexus, the Resi Digital Conference Series, the Dana-Farber Harvard Cancer Center, the Child Brain Tumor Tissue Consortium, and then the NF Open Science Initiative, the NF Registry, and the NF Data Portal. Thank you so much. Without you, of course, we wouldn't have the resources to pull this hackathon off. And then our incredible community partners that helped us to spread the word, get people involved, and register. I will not read them all, but here, thank you so much for all of your support in providing mentorship, providing the right participants to take part into this amazing event. And of course, special thanks also to all our mentors, experts, and our experts of the condition. So our patients mentor. There's an incredible pool of talent. We are so grateful. Without you, we couldn't really create this event. So thank you for all of your involvement, all the time that you dedicated to this over the 6 weeks. As well as our organizers and judges. So the judge will start working right today, looking at your project and reviewing them. And we have an incredible lineup of talent there and expertise as well, that I'm sure they will be the best reviewers for your project. And one final thank to Be My App, our partner and platform support for all of the logistics and managing. So Lucas and Zach, thank you so much for incredible time and support that you gave us. And last but not least, thank you to the audience and to this over 600 registrants and more than 20 teams that participated in the Hack4NF. It's been incredible, the response to this type of events, and we'll hope to continue this in the future and evolve this even more. Thank you so much for dedicating your time and talent to NF. And you definitely have joined the fight that we will not stop fighting until the very end, when this disease is going to be eradicated as we know it. So after this, I will turn it to Lucas. And let me stop sharing my screen, and we can start with the project playing.
Awesome. Thanks, Salvo. And thank you again to everyone that participated and sent me your presentations over the last couple of days. Without further ado, I will go ahead and start sharing my screen. And we will get going here first with Team Pharmacom.
Hi everyone, I am Sharavana, a biomedical researcher with University of Pennsylvania, presenting for Team Pharmacom. Our goal is to accelerate drug discovery and drug repurposing against NF1 and NF2 by providing access to high-throughput drug screen data to researchers. Screening generates thousands of data points, difficult to read even to the researchers who work with them. Team Pharmacom developed an interactive website to comb drug screening data, untangling the pharmacology of NF1 and NF2. This web model is similar to a zoomable street map, where you zoom out to see where an address is in the city or zoom in to see the cross streets or the building. Our website, a temporary link shown in subtitle, allows you to zoom in, zoom out, or search through over 10,000 data points. The heat map has drugs organized based on pathways on the left side against NF1, NF2, mouse and rat cells on the right side. The data is presented as color-based scale of IC50s for each drug. Strong drug or cell combinations are blue and weak ones are red. Uniquely, hovering over a colored box shows up absolute IC50. You can zoom closer by clicking the colored box to reveal the dose response curve, IC50 again, and health slope. Search boxes on the top filters your view to drugs, targets, or pathways. For example, typing cell cycle will bring up these drugs. MapK brings up these drugs. Clinically tested drugs for neurofibromas like sorafenib showing mostly red, predicting its failure in clinical trials against neurofibromas. MEK inhibitors brings out this blue logo. MEK inhibitors are the best drugs that distinguish NF1 from NF2, as NF2 cells respond poorly to them. Interestingly, a recent FDA-approved MEK inhibitor, selumetinib, is not a particularly potent MEK inhibitor, suggesting that other MEK inhibitors may be more effective. Intriguingly, the preclinical screening has been a good indicator of neurofibroma responses, even though testing was done in MPNST. These types of data have some limitations to keep in mind. Cells used are grown in vitro and not in vivo, where they have a different microenvironment. Additionally, some drugs need prior activation. For example, ifosfamide, a drug recommended for treating MPNSTs, shows no activity as it requires activation in liver. Nevertheless, Pharmacom allows a rapid comparison of drugs, targets, and pathways against NF1 and NF2 cells. We are refining our website by adding new filters to select single or multiple pathways, targets, or drugs, and also a PubChem link to each drug. We will integrate Pharmacom with website at the University of Pennsylvania along with detailed methodology. We hope to add the Synodus data on neurofibromas, but the Synodus data is in a different format and converting is very labor intensive. With time, we also hope to include genetic information into the website. We are a team of 8 members and who developed this website. Veronica, Elise, Nancy, myself have worked independently to generate the data points and process the primary data. Aaron from Genentech, who did coding to add filters and search options, and Wendy from NCI, who developed algorithms for curve fitting. Dr. Jeffrey Field advised our team. Thank you all for your time.
Awesome, and thank you to Team Pharmacom. So as a reminder, everyone, if you have any questions for the team, please feel free to put them in the chat. And I will give everyone a few moments here to answer those questions, or to ask those questions rather, and then we will get started with the next presentation in just a moment. This will be self-updating causal models to accelerate discovery in NF.
Hello, my name is John Bachman, and I'm presenting for a research team from Harvard Medical School that I co-lead along with Dr. Benjamin Guri. I'm also a family member of an NF1 patient. So our submission falls under the drugs and targets focus area, and our goal is to help researchers to make sense of new discoveries in NF with a focus on the molecular mechanisms that are relevant for drug discovery. We want to help scientists to get a comprehensive picture of what's already known about NF and how each new finding fits into that existing knowledge. So our approach is to use text mining to read new scientific publications every day, update NF-specific causal models, and use those models to make connections, generate hypotheses, and highlight scientific novelty. Our submission consists of a platform called EMMA, which you can browse at emmaa.indra.bio, and 2 causal models of NF contained in that platform that together can help NF researchers accelerate the discovery process. So the first model is built directly from text mining the 18,000 PubMed articles about NF, and it contains about 7,000 statements about the functions and interactions of NF1, NF2, and other entities mentioned in those articles. The network can be browsed and queried using the index network sharing site or explored in the EMMA web application, where the supporting evidence behind every statement can be viewed, along with links back to source publications. And if you're logged in, text mining errors can actually also be curated to improve the system over time. So the key feature of this approach is that the model is not static. We process new articles appearing in PubMed every day and add new relevant statements to the model when available. NF researchers can subscribe to updates to the model by registering causal queries on the site and getting updates by email, and also by following the EMMA NF model on Twitter as well. So this continuously expanding NF knowledge network is one part of our submission. The other part is a manually curated causal model of the RAS, NF1, and NF2 pathways that we're in the process of building right now. The model is completely transparent even for non-modelers because it's built from about 200 simple declarative English sentences. And the model is listed on the EMMA site as the RAS model. And the key thing here is that we can use this detailed model to explain the causal claims that we find in the literature-derived NF knowledge network. So for example, from text mining, we find a number of these claims that we can check against the detailed model. And one of them is that NF2 inhibits PAC1, for example, from these sentences. And we might want to know mechanistically how this occurs so that we could understand how to intervene with a drug. And using the detailed model, we can find that this can be explained by a causal path showing that NF2 competes angiomotin away from inhibiting ARH-GAP17, causing inhibition of CDC42, which would otherwise activate PAC1. So conversely, when there's a new empirical finding that's not explained by our detailed model, that tells us that that finding is mechanistically novel or that our model needs to be improved. So importantly, in this platform, any set of causal relations can be used to explain any other set. So we could use either of these models to generate hypotheses to explain, for example, why certain drugs appear to work in an in vitro drug screen. So we are excited to share this work with the NF community and feel that these resources could become hubs for assembling actionable knowledge about NF for drug discovery, and in the future for other rare diseases as well. Thank you very much.
Awesome. And thank you to Benjamin and his team. I'll give a little more time for questions to be asked in the chat, and then we will get started with our next presentation, the NF Care app, in just a moment.
Yeah, thank you. Maybe something we can do is if someone from that team is available to answer a question during your team presentation, please type a message and say, I'm here, I'm available to answer questions online. So perfect. Yes, thank you, John. So that people knows that you are able to answer questions. So please just type your questions and John will be able to do so. And during next presentation, some members from the other teams can do the same and move forward. Thank you.
Awesome. And like I said, we will go ahead and get started with the NF Care app.
The NF Care app is an app for general practitioners caring for patients with NF. There are 66 clinics in the NF Clinic network caring for 125,000 Americans with neurofibromatosis. Neurofibromatosis is a rare disease that many practitioners aren't familiar with, and it affects many different body symptoms with a wide range of symptoms. There are few specialists, and the few locations mean that there is a true barrier to care. One patient we spoke to, William Ritter, said that it took him 25 years to be diagnosed from the time of the first tumor removal. Our solution is the NF Care app. The audience is primary care doctors, pediatricians, family medicine, general practitioners, residents, and even medical students who need to learn the key diagnostic criteria, view management recommendations, treatments, see referral guidelines for specialists, and provide the long-term care and surveillance for patients with NF. Let's take a look through the app now with Bill, one of our team members.
The app is broken down into 3 sections. First, we have the diagnostic criteria. It is broken down as a list of symptoms. There is an I button for clinicians to view more information about each symptom. Then they select which ones are relevant to their patient. Once a diagnosis is confirmed, they can review the care plan and treatment page, which includes the annual visit information. This is filterable by age so they can review infant, child, teen, or adult annual exams, as well as anticipatory guidance for future planning. Further on, we have the symptoms broken down so that practitioners can view manifestations. For each manifestation, they can view the definition, care plan, treatment options, and when to refer to a specialist. All of the information in the app is referenced thoroughly so that in the resources section, practitioners can review the literature, publications, sample images, and view tools for further study.
Our team consists of Bill and myself, as well as Patrick Childers and Dr. David Wolf, who's a pediatric neurologist and an NF1 specialist. The next steps for this app include building out the NF2 and the Schwan neurofibromatosis diagnosis and care plan pages, including additional resources and securing the copyright use for the resources, reviewing all the content, and having it certified by a medical team. In the meantime, we invite you to download it at the Test Flight app and view the app for yourself. We welcome your feedback.
Awesome. Thank you, Lauren and your team. I'll do the same and give a few moments for questions to be asked. I know Lauren is in the chat to answer any questions if anyone has any. And we will get started with Schwan cell development here in a moment.
Hello, everyone. My name is Xiyuan Zhang.
And I'm Vishaka Gopalan.
We're from the National Cancer Institute. Our team studies the Schwan cell development. We hope to use our analysis to provide you a brand new angle to understand the origin of this disease from a developmental perspective. NF1 associated plexiform neurofibroma develops when there's complete loss of the tumor suppressor gene NF1 in the Schwan cells. These benign tumors can undergo malignant transformation to form MPNS T, which often exhibit dedifferentiated phenotype. We're interested to understand if the neoplastic Schwan cells from benign plexiform neurofibroma and MPNS T are showing the same stem cell feature. To do so, we took an unique approach by using the single-cell RNA sequencing data of developing Schwan cells that are publicly available. As illustrated here, Schwan cells originate from neurocrest stem cells through Schwan cell precursor to become mature myelinating or non-myelinating Schwan cells. The usage of single-cell sequencing allows us to build a transcriptional trajectory that is perfectly aligned with the development of normal Schwan cell.
Yeah, this trajectory was constructed by integrating data sets from mouse embryos at 2 stages and from an adult mouse. Now we know that such integration can introduce batch effects that can overpower any biological variation. But we were able to minimize these effects, and we're certain that our trajectory is correct since the Schwan cell precursors come from the correct pool of cells in the mouse neurocrest. Next, we use this trajectory to derive signatures for each of the clusters and use them to evaluate which embryonic cell type is being recapitulated by Schwan cells in plexiform neurofibroma. And we then use the human orthologs of these signatures to score bulk RNA-seq profiles of human MPNST tumor samples, which allows us to evaluate the extent of dedifferentiation in these tumors.
Here, we built a normal Schwan cell development trajectory and projected Schwan cells from plexiform neurofibroma and MPNST onto this trajectory. Our results show that neoplastic Schwan cells from benign tumors exhibit malignant myelinating Schwan cell feature, whereas the malignant cells from MPNST behave more like dedifferentiated Schwan cell precursors. We believe that our analysis of the single-cell RNA-seq data of developing Schwan cell will benefit researchers in the NF research community to better understand the cause of the disease. And we hope to help NF patients by preventing the malignant transformation. Thank you so much.
Thank you.
Awesome. Thank you to the Schwan cell development team. If anyone has any questions for them, they are in the chat. So be sure to write your questions down and direct them to them. I'm sure they'd be happy to answer. And in just a moment, we'll get started with our 5th presentation, Pocket Audiologist.
Greetings everyone, this is Vishwanath presenting on behalf of Pocket Audiologist crew. So this project mainly deals with developing an smartphone based application for the NF affected individuals. So what are the key benefits that we provide with the help of our app is rapid diagnosis, telemonitoring, hearing aid check, mobile application, smart therapy and biofeedback based sessions. So how we can use this application is that the user has to open this app, listen to the sample audio tracks and try to repeat the same and record it. And within a few seconds, our application will provide scores like how well he or she has perceived the audio track. And to develop the proof of concept for this application, we have used LabVIEW software to develop the concept prototype. And we will now have a walkthrough over the developed prototype. This is the user interface for the application that we have developed. We have multiple panels here. We will visit one by one. First up, we have the configuration panel, wherein the user has to listen to the sample track, and based upon his audiologist suggestion, he or she can fix up his own parameters and save the configuration. Once it is done, we have to shift to the test panel, wherein we will be having audio tracks for the user to listen, and he or she can play the sentence and listen to the track and record the same. Once the recording is over, then they can check the results by clicking on the respective button, and we will have a quick notification over the scores being generated. So this platform runs on a speech processing based technique, which is uniquely designed for the processing. And correspondingly, we have another panel, which is the in-depth results, which is shown as per the user's interpretation of the sentence being listened. These are the important USPs that we offer to our users. First, instant evaluation and access from anywhere with privacy. And next, requires no complex gadgets, very intuitive and simple UI, which is very much evident from the tool walkthrough. And we have also planned a set of activities to scale up the entire project as a phase 2. First up, we will be validating the tool's effectiveness, and we will be designing the contents, the learning contents based on the audiologist suggestion. And we also have plans to develop it as a large scale application for Android and iOS mobile phones. And the technologies that we have planned to use are Google Assistant, cloud computing, speech processing, and data analytics. Apart from me, Praveen Kumar Ji and Om Prakash S has supported and played a crucial role in this project.
Thank you.
Awesome. Thank you to Vishwanath and the Pocket Audiologist team. He is in the chat if anyone has any questions. And in a moment, we will get started with gene networks-based drug discovery in plexiform neurofibroma. So if anyone from the team is watching the webinar, be sure to say hi in the chat so folks can ask questions, and we'll get going in just a moment.
Hello, everyone. My name is Dao Chun Sun. I'm from the team at Terminator to present our project, Gene Regulation Network-based drug discovery in plexiform neurofibroma. A drug screening is really needed in the NF1 field because the patient and the clinicians really have two less options to treat these tumors. However, after the drug screening using this 20,000 small chemical library, we suddenly find we have too many candidates. What we really want is the best one, or at least ranked candidates to prioritize. Well, at the same time, we have to keep in mind that the candidates in the drug screening have very low successful rate on the real tumor. The scientists will ask, does this efficacy have a reliable biology to support? And the clinicians may be interested in combining different candidates to enhance the response. We devised this magic strategy using R. We create the gene regulation network from the cells using the screening and also the patient's primary tumor. to get the preserved gene regulation network. At the same time, we filter the drug response data and calculate the correlation between the drug response and the preserved gene regulation network. And then use that result, we separate the drugs into different clusters. In each cluster, you will have some well-annotated drug and also the novel one. Use annotated drug targets, we can calculate the enriched gene ontology term. And in the same way, we also can get those geoterms from the preserved gene regulation network. Then we calculate the geosimilarities between those gene network and the drugs cluster. This is very important steps here to reinforce the biology. And then we end a very surprising list with the sunitinib ranked the first. This is really big bingo because this is the first FDA-approved platform neurofibroma drug. Why our strategy is great, I think we use the preserved network, which enriched the candidates targeting both cells and the tumors. And the algorithm weight higher on those well-annotated drug candidates. And use the gene regulation network and the drug clusters can help the researchers to nail down the mechanism and also identify the novel candidates. And the drugs from different clusters can help the clinicians to choose combination. I want to acknowledge all the people who provide support to this study, and also want to remind everyone that plexiform neurofibroma is a young people disease, and the awareness and understanding of this disease from the young generation really makes sense different. So this is my 5-year-old daughter's strategy to ending the NF. Thank you very much.
Awesome. Thank you, Dao-Chun. We'll get started with our next presentation here in a moment. Our next presentation will be AI for NF tumor detection. So if anyone from the team is here, just a reminder to say hi in the chat, and I will get this playing in just a moment.
Hi, I'm Isaac. I'm part of Team Schwan, and I'm going to talk about our project using deep learning to detect NF tumors on MRIs. For more detail, you can see our project page, and this is our contact info if you'd like to get in touch. So the technical term for what we're doing is tumor segmentation, which just means marking tumors on images. And we investigated an artificial intelligence approach, specifically a type of AI called deep learning, based on mentor feedback that would be interesting. Why deep learning might be good for this? Deep learning has revolutionized many image-related tasks in recent years. Think self-driving cars, for example. It's achieved record-breaking computer performance, in some cases even surpassing human experts. NF tumors might be particularly hard to detect because they can occur in large numbers in many different places on the body, different sizes and shapes. So a good automated method could help detect and track them. Why deep learning might be hard to use for this? A small amount of data to learn from. Deep learning is powerful, but typically needs a lot of data. And it's a relatively complicated task. As we said, tumors can be in many places, sizes, and shapes. That makes it harder to learn. And it's also unbalanced. Less than 1% of the pixels are tumor positive, which is also going to make it harder to learn. So we used a variety of methods to try to use deep learning anyway. And we got off to a decent start. F1 score of 0.62. F1 score is a standard method used to measure unbalanced tasks like this. And we'd like the opportunity to try some further improvements. So we used a variety of methods, including image processing, histogram equalization, and contrast enhancement. We used UNet, which is pretty much the go-to deep learning architecture for this kind of task. But we used it with something called transfer learning, which helps you use deep learning with a lot less data to learn on. And finally, we generated some extra training examples by slightly rotating the MRI image stack in all 3 dimensions. And as we said, we got some decent preliminary results. And we would like the opportunity to try some further improvements, particularly one that's based on something called in-painting, which we think can significantly improve performance. Thank you for your time.
Awesome. Thank you to the NF tumor detection team. I know Lars is in the chat. If anyone has any questions for him, I know he would be happy to answer them. And up next, we have Malignant, machine learning tool for NF1 gene analysis.
Hi, I'm Kevin Bremer, and I'll be presenting the project I've been working on with Nicholas Spies entitled Malignant, Machine Learning to Identify Gene Novelty Across Neurofibromatosis Tumors. Malignant is a random forest machine learning classifier, where we take the RNA-seq dataset and we classify the data as stemming from either a benign NF tumor or a malignant NF tumor. This allows us to create a training dataset that then the computer can make a series of decisions at nodes that represent 4 genes. If these genes allow the computer to better classify benign and malignant tumors, it will make a positive decision, iterate this process, and come at a prediction, which are then combined into one algorithm, and then we can use that algorithm to identify novel gene targets that distinguish benign and malignant NF tumors. We did 2 searches based on the Malignant platform. One is an unbiased total genome search, where we identify 3 genes that are important in kinase signaling that you can further read about in our project report. We also identified uncharacterized genes that are only available in the Human Genome Project. We then did a more specific search, which looked at kinase genes, which are important in neurofibromin's upstream signaling of the kinase pathway, as well as glycosylation genes, as I'm involved in a wet lab project sponsored by CTF in order to study glycosylation in neurofibromatosis. We identified 10 genes that are interesting between benign and malignant tumors in both of these pathways. We also did traditional RNA-seq search between benign and malignant tumor types of NF. And the example shown here is the difference between a plexiform neurofibroma and an MPNST tumor type, where the genes represented on the right of this graph are more highly expressed in malignant tumors, and the genes represented on the left of the graph are lower expressed in malignant tumors. And this will hopefully allow us to identify novel gene targets between both benign and malignant tumors from 2 different analyses through the Malignant platform, as well as traditional RNA-seq. We also looked at drug cell viability data provided in the hackathon and found that 5 of our malignant kinase genes had available drug target data. And interestingly, dovitinib hit 3 of our kinases, CDK6, NEX6, and PKA1, suggesting that this drug may be interesting therapeutic potential for NF. And a recent report also found that dovitinib has therapeutic potential in a cell model of NF2. So hopefully this shows the power of Malignant as well as RNA-seq analysis in identifying novel gene targets for therapeutic intervention of NF. If you want to learn more, please visit our project page. We have the GitHub link to our Malignant code, as well as the RNA-seq Excel files, and a project report with a further analysis on these gene targets. And we'd just like to thank the organizers of the CTF Hackathon for a great 6 weeks.
Awesome. Thank you to Kevin. I know Kevin is in the chat as well. As a reminder, if you have any questions for him, be sure to drop them in the chat. And without further ado, we're at our halfway point with our 9th presentation. This is CNF drug discovery, copper and zinc as new candidates.
Hi, I'm Isaac. I'm part of Team Schwan. I'm going to talk about our project proposing copper and zinc complexes as new drug therapies for CNF. For more detail, you can look at our project page, and this is our contact information. So CNF is a tumor very common in NF1. It can be disfiguring and painful, as you can see. In fact, it's considered the most burdensome feature of NF1, and there are no approved drugs. We combined gene expression data, the Synapse Drug Target Explorer, and a literature search to come up with some possible new drug candidates. To sum up, we found that many of the most overexpressed proteins in CNF tumor tissue are copper or zinc binding, and these proteins are also important in the natural tumor suppression and immune anti-tumor response. So our hypothesis is that this overexpression is part of the natural protective anti-tumor system, and since they are highly overexpressed, their activity might be limited by the availability of copper or zinc. So raising cellular copper or zinc levels might make the anti-tumor response stronger. So therefore, we propose to test copper and zinc complexes against CNF in the lab. Our methods, we had 3 steps. We used Z-score values to compare gene expression levels between samples. We compared 33 CNF tumor tissue specimens to 2 normal cell lines, Schwan cells. We found the following 10 genes as most overexpressed. This is a heat map giving more detail on that, showing the level of overexpression is high for all of them. We checked the Drug Target Explorer and found that most of these genes coded for copper or zinc binding proteins. Finally, we did a literature search and found all the copper binding proteins are important in tumor suppression or the immune anti-tumor response. Going down the list, apolipoprotein D inhibits cell growth and is a direct target of likely tumor suppressor P63 and P73. Gelsolin upregulation reduces invasiveness and suppresses cell proliferation in several cancer cell lines, and lower expression is correlated with poor survival. KRT10 and KRT1 is part of the cytoskeleton, but it's also known to regulate cell growth and apoptosis. KRT10 in particular, high expression inhibits cell proliferation and prevents tumorigenesis. And beta-2 microglobulin, a key immune system protein that's known to be important in the immune anti-tumor response. Finally, this is a list of some of the known compounds, including anti-inflammatories and a possible transdermal compound that we'd like to test in the lab. Thank you for your time.
Awesome. Thank you to Isaac and Lars. They are both in the chat again. So if you have any questions for them, be sure to drop it in the chat. And like I said, that was our halfway point of presentations. So we're moving on to number 10, which is NFPH, which stands for NF Personalized Health. And as a reminder, if anyone from the NF Personalized Health team is in the webinar, be sure to say hi in the chat so that folks can ask questions.
NF Personalized Health, a neurofibromatosis symptom tracker.
When meeting with a patient mentor, NF2, we asked her what her concerns were when addressing the current resources available. Of the concerns, the most pertinent problems included keeping track of the symptoms and being able to remember which provider to share these details with. Our patient mentor currently uses a note-taking app. However, this leads to inconsistencies and incomplete notes. Along with symptom tracking, we wanted a pragmatic way to incorporate testing at home due to the limited available clinic resources for NF. Our patient mentor suggested testing hearing loss as one of the most important changes to track.
We built an app specific to NF symptom tracking plus a built-in hearing test, so users can have an at-home resource to measure hearing amplitude progression over time. Additionally, we aim to open the platform to providers, giving them real-time update and allowing them to quickly share feedback with their patients.
Overall, this solution provides users their own specialized entry logging system and a platform for tracking changes in hearing, skin spots, and other symptoms. This app can help build stronger relationships between doctors and patients, and if users opt in sharing their symptoms, this can further support research in recognizing and treating neurofibromatosis.
Thanks from our team, Iona, Scott, Carly, and me, Candice. All of us are invested in building a better future, and we enjoy taking this opportunity to work together to contribute towards better NF care and quality. We hope to see you again in the future, and thank you for taking the time to learn about our project.
Awesome. Thank you to Candice and the NF Personalized Health team. Candice is in the chat as a reminder if anyone has any questions for her. Our next presentation, number 11 in our lineup, will be the MyNF team. And we'll get started in just a moment. If anyone from that team is in the webinar, just another reminder to be sure to say hi in the chat, and we'll get started in a moment.
Hi, we are Team MyNF, and we are a bunch of technologists coming at you from across the globe. The way that we wanted to approach this hackathon was by better understanding the challenges that patients and caregivers face on a daily basis. And so we spent the first couple of weeks of the hackathon conducting interviews with patients, caregivers, doctors, and researchers in the NF community to better understand those challenges. One of the common threads that we found is that patients are interacting with a multitude of different specialists and providers. And if they don't attend a specialized NF clinic or institution or don't have access to one, then typically the burden of responsibility of coordinating care as well as information exchange between all of those providers falls on them. We tried to quantify this challenge by sending out a survey, got back a lot of great information, but of note, participants on average were interacting with 6 or more different doctors in the past 2 years, and one participant was actually interacting with 15 different specialists. The problem that we're trying to solve is that patients with NF can have many, many different providers, and so we'd like to alleviate the burden of coordinating information exchange between those physicians. The solution that we ended up coming up with was an application that empowers patients to keep track of their medical journey and allows patients and caregivers to record their medical data and then share it with providers. So we've designed an application that's mobile friendly so patients can carry their information on their smartphone from physician to physician. After you end up logging in, the first screen that you end up seeing is a chronological timeline of all the appointments that that patient has had. You can click into each of those appointments and get more detailed information on what was said during that session, as well as view any images or scans that are related to that particular session. We also heard that medications are pretty difficult to keep track of and are important to accurately give that information to other providers. And so you can also, in addition to appointment objects, add medication objects where you keep track of information such as the medication name, dosing, and frequency. So we've ended up building a prototype over the duration of this hackathon, but this isn't the end for MyNF. What we would like to do is put this prototype in the hands of patients and caregivers and really understand what is the value of an application like this in easing the burden for storing medical information by conducting a survey. And so we believe that by putting this in a research framework, an experimental framework, that we can validate that an application like this is useful, as well as help to mitigate some of the regulatory requirements that are required for storing patient information. In addition, there are other directions that we could go after this analysis, such as giving patients the opportunity to share their data with researchers, and then also providing an API-based way to share information with providers at major medical institutions. That's the end of our presentation. Thanks so much for listening to all of our mentors who shared their expertise on neurofibromatosis, as well as the Children's Tumor Foundation for throwing this hackathon.
Awesome. Thank you to Matthew and the MyNF team. Matthew is in the chat if anyone has any questions for him. And without further ado, we will move on to number 12, which is AVI, a virtual pet for children with NF. So if anyone from the team would like to say hi in the chat, please feel free to do so. And I'll give everyone just a few moments here. I know we've been flying through these presentations a little quickly, so I'll give everyone a couple of moments before I start this next one. And Enrique is in the chat for this next presentation, the AVI team. So without further ado, we'll get started.
Hello, and welcome to our presentation about our application, AVI, a virtual pet for children with NF1. This is our team. We have 2 data scientists, 2 software engineers, and a designer. Let's talk about the problems first. There are too many symptoms in NF1 to effectively collect longitudinal data. Parents are inundated with information to keep track about their child's condition. Keeping track of symptoms is tedious, boring, and confusing for kids with NF1. And parents don't have a positive and easy way to talk to their child about their disease. We've created a solution for kids diagnosed with NF1 to track their symptoms using a fun and positive interface while gathering data using NLP and image classification. There are many benefits. We know that kids with this disease are going to be surrounded by somber messaging about the quality of life. And so we set out to create a more engaging and optimistic experience. The more the children understand about their body, the broader vocabulary they have to communicate about their condition. Users of this application will have a new helpful tool to advocate for child's treatment in doctor's appointments. In longer term, the app will provide a central archive for children's families to store all information surrounding symptoms, MRI imaging, images of skin marks, bumps, diagnosis, details, and more. And finally, we're collecting data using NLP and image classification to inform users of other possible complications, as well as track existing symptoms. And this is helping the NF research community. How will we do this? Well, meet AVI. AVI serves as a companion to help incentivize children to communicate their feelings and about their feelings and symptoms. As kids log entries consistently, AVI grows and thrives. AVI will develop brighter fur, new facial expressions, and more animation. If the log is neglected, AVI decreases in size and vibrancy. It's the child's responsibility to care for AVI and in turn themselves. By using natural language processing model, we are able to extract crucial data without much of the user's engagement cost. Asking a simple question like, how are you feeling? We can identify NF1 symptom categories from text log and store them for easy data collection. Then using the analysis of all patient data, we inform other likely complications to look out for. Symptoms can appear visible on the skin in many different ways. It's hard for parents and guardians to keep track of all skin lesions over many years because it could be hundreds of them and they appear at different ages. We use image recognition to make the process easier. Whenever the parent uploads a picture, we classify the lesions, keeping their photos, numbers, and sizes so that the parents have an archive of their children's skin lesion history as they grow up. Thank you so much for your attention and your consideration.
Awesome. Thank you to the AVI team. Like I said earlier, I know Enrique is in the chat and he would be happy to answer any of your questions. I'll give everyone a few moments and then we will move on to our next project, which is NF Knowledge Explorer.
Hi, everyone. This is the pitch for the NF Knowledge Explorer project from the Curate team. NF patients, healthcare providers, and NF specialists have a lot of questions. And the questions you see here may really come from all of them. Do NF1 patients have precocious puberty? Is growth hormone imbalance related to NF1? Or what are all the reported phenotypes for a specific mutation? The problems here are there is an increasing amount of literature. A PubMed search for neurofibromatosis retrieves 18,000 papers. The literature is often behind paywalls, and even when it is available, it is hard to navigate. Not many patients go to PubMed, and Google does not search full-text copyrighted articles, which is where the mutations are mentioned. Moreover, doctors are busy, patients are busy, and they may read only key articles, and this introduces a bias. Our goal is to facilitate knowledge discovery by aggregating insight from different sources, and JP is going to talk about that in detail.
Here is the overview of our implementation design. There are three main components. First, we collected data from different sources in order to curate a better knowledge base. Second, we applied the state-of-the-art language model for the question answering task and served the model results through an API in an interactive UI. Lastly, we want to incorporate a feedback loop mechanism which allows volunteers with domain knowledge to validate and annotate useful information to improve the knowledge base and model. Now, let me demo the interactive tool. First, a patient might want to search for specific mutations, and then we can see the aggregated insights related to the diagnostics and symptoms. And here we can also show the details from the ClinVar mutation datasets. Now, we can continue to ask a specific question related to the mutations or the symptoms. And here we will provide the most relevant answers from the literature data. And here it says the answers are relevant, but we definitely want to continually validate the results with subject matter experts. So that's why that's also part of the reason why we want to create an annotation tool. We think a feedback loop is important to not only continuously improve the customized model, but provide opportunities for the community to validate information and curate knowledge together. Here's the example documentation we curated in a FAQ format. We think it could be useful to put on the website. To summarize, our goal is to facilitate NF knowledge discovery through prototyping the end-to-end process from data collection to knowledge curation. For next steps, we want to collect feedback from the community to better understand the pain points, especially when seeking for information and knowledge discovery. Thanks.
Thank you to the NF Knowledge Explorer team. As a reminder, if anyone from the team is in the chat, be sure to say hi so folks can ask questions. And up next, we have the Viometrics team. So getting down to the last few presentations here. So be sure to say hello in the chat and we will get started.
Viometrics is a project that we put together using 3D scanning combined with video game algorithms. Our daughter has a plexiform tumor in her left jaw that extends into her neck and eye. It never has had clear margins on MRI. Being able to take a picture and see the difference in her tumor growth over the years would be a game changer in her care. Enter Viometrics. It's a way to take 3D scanning technologies and count tumors and calculate volumes. MRIs are still the cornerstone of care, but Viometrics can be an alternative method to obtain metrics through photometric scans. This can be done in clinic or on a more regular basis, monthly, even weekly. It's easy to track over time and has the ability to detect small changes. Our team consists of myself and Patrick Childers. We have a background in video games as well as medical software. The overall process is really simple. We're going to take a 3D model of the arm, and then we're going to identify the tumors, isolate them, and calculate their volume. For our experiment, we're using modeling putty to simulate cutaneous neurofibromatosis. Patrick is going to walk us through how the 3D modeling works.
For capturing the 3D model itself, we looked at both the newer versions of the iPhone that contain the true depth cameras as well as professional 3D scanners. We found the iPhone's cameras to be lacking in the resolution we needed to perform the calculations.
The professional cameras, on the other hand, captured much more detailed and accurate measurements. The professional scanner we used was made by Polyga, and they were generous enough to let us use one for this project. The scanner itself uses structured light scanning. Structured light scanning uses light patterns projected onto the surface of an object. Stereoscopic cameras then record the surface of the object by measuring how the light deforms around it. This allows the scanner to create a highly accurate 3D model that we can then load into our software and run our algorithm analysis.
Once we have our 3D model, we can use graphics algorithms to identify abnormal skin curvatures. We're looking at edge angles and surface normals to find the areas of the skin that are different. Once those abnormalities are identified, we can isolate them into bounding boxes. Then we perform a volumetric calculation to get a volume for each of the tumors. Now we took a single tumor from our experiment, getting fairly close to our expected measurement for this proof of concept. More testing and calibration will be needed to be performed in the future. We believe that this can be used with plexiform neurofibromatosis in connection with MRI and CT results to provide relative comparisons over time between plexiforms. We also believe this technology can be expanded and used in other areas, and we look forward to seeing how this technology will evolve over time. Thank you.
Thank you to Lauren and Patrick. I know they are in the chat as well if anyone has any questions for them. Up next is the NF Matrix team. So I know you've heard me say it a lot, but don't be afraid to say hi, and we will get going in just a moment.
Welcome to the Matrix. We would like to introduce you to a rare disease platform built specifically to help the many needs within the NF community. The Matrix is meant to connect the dots and data across NF patients, caregivers, healthcare providers, and researchers. The first perspective we wanted to capture is the patients or caregivers. As you can see, the system is set up to capture rich data such as demographics, medications to be used for adherence and alerting, symptoms and activity tracking, as well as correlation through insights. Narrative text can be captured in our journaling module. Messaging to patients and caregivers can be performed through our messaging module. Informed consent and structured data capture for clinical research and clinical trials can be submitted through our forms. Any type of document can be uploaded by the patient or caregiver, and then all the data can be shared through our sharing center. Finally, NF-specific resources and materials are available through our resource center. As an NF healthcare provider, use the Matrix platform to view and observe real-time data from your NF patients. The final perspective we will cover is from the researcher perspective. As you can see, we have geographic maps that show patient populations across the world. We have messaging that allows specific messages to go to groups of patients or individual patients. Lastly, you will see a robust form builder that can be used to structure natural history studies, registries, or other clinical trial data capture form. The platform is web-based and uses a responsive design so that it can be used on mobile devices as well as tablets with no issues. The platform was built with the end in mind, leveraging all the necessary regulatory compliance. This is our team. We were humbled and blessed to speak to over 12 mentors. We tried to bridge and blend the insights we gathered and deliver a robust platform to help the various needs we heard. We feel the NF Matrix platform is a solid foundation in which to meet many current needs, but also provide ability to add on to in years to come. This is an amazing community, and we hope we are selected and can make a difference through our work.
Awesome. Thank you to the NF Matrix team. I know Jason is in the chat if anyone has any questions for him. We're coming down to the last few presentations here. Up next is Central Point. So be sure to say hi in the chat. There you go, Kay. I just see you just said hello. So if you have any questions for Kay, please feel free to ask and we'll get started in a moment.
Hi everybody, I'm Kate Ashton, MBA, Patient and Project Manager in the team Central Point. As well as myself in Central Point, we also have Roland, Anna, Gustavo, Ariel, Joel, Jan, and it's been funded by GoBoo.io. We have built Central Point for a way for you to track your NF-related problems. This can be from pain to cafe au lait markings in a fun and engaging way.
Medics don't know enough about NF to start living with it. There's no central location where patients can track problems. This prevents medics and researchers to easily access data. The main challenge is how to provide a simple and funny way to record it and a much more efficient interface for medics and scientists to visualize data. We propose 3 main points: a companion app, knowledge graph, and a joint vision. Patients, medics, and researchers having a better view of the different dimensions of NF. We rely on an NLP-based pipeline to ingest data from documents in the knowledge graph with an interface for chatbot that will talk with patients and other users. We got the input from questionnaires sent to NF community, input from meetings with mentors, and data sources from Hackathon. We highlight as results the knowledge graph visualizer that will allow anyone to access data and better understand the relation between different entities, and a cross-platform NF chatbot that will communicate as a human being.
Today, we'll be demonstrating what the Raspberry Pi would look like as part of a smart mirror, and it is also going to be available on iOS devices as well as Android.
Hi there, my name's Julie, and I'm Kay's mum. I'm using this to track my pain and how my NF tumours and cafe au lait marks are going. I am in pain today. 9. I have also found another cafe au lait mark on my leg. I have found another NF tumour on my arm.
We got some very good achievements so far. What is our roadmap? The potential of knowledge graph, NLP, and deep learning is huge. We envision a crowdsourced project with contributions from all the NF community, a publicly available project at GitHub to make this concept grow. In the meantime, we have learned a lot.
Muchas gracias. Adiós.
Muchas gracias. Adiós.
Adiós, gracias.
Muchas gracias. Hasta la vista.
Adiós. Muchas gracias.
Thank you. Bye.
Awesome. Well done, Kay and the Central Point team. As a reminder, if you have any questions, I think Kay and her entire team are in the chat. So be sure to ping her there if you have any questions. Down to our last 3 presentations. Up next is Therapeutic Wellbeing. We'll get started here in just a second.
I'm founder of Cirrus Cloud, and today we are presenting the solution of Hive. Hive is an enterprise intelligence SaaS company for the healthcare industry, which we do for patient engagement. What we did is today the problem is the patients who are having skin diseases like psoriasis or anything. would find difficult to get information online or get motivated by the medicines prescribed by the doctors or well-being. So there's no further human beings where they take care of the patient. So what we did is we developed a chatbot engine which we master the patients, and we make them participate in many motivation programs and well-being, and we will set a goal for achieving the purpose. So this is the so we did a chatbot which is built on artificial intelligence, machine learning, and natural language processing. That means patients can ask very basic questions, and the system would understand based on the healthcare industry standards, and would provide them suggestions and information wherever required. And the demo for the portal is this one. See, this is a Hive chatbot where the chatbot is asking the patient's questions, and chatbot can answer, and subsequently the chatbot can answer questions. So if you see here, here patient is asking what is a combination of this is here, and the chatbot is answering a few questions on the psoriasis skin cases. So this can be applied across many verticals across healthcare industry. Okay. So going back, business model, we are offering pay-as-you-go or platform subscription model to the health industry. On the traction, we already developed application for GS where we got selected from top 10 in 2018 and Barclays Labs. And the competitors, we are charging less charges. For example, we are charging $6 per user or per 1,000 charge, whereas the competitors are €358 user for first charge. My team is based in London. I'm the founder. I did my master's, worked for many health companies as in the implementation and as the co-founder. Venkat is.
Well done. Thank you to the Therapeutic Wellbeing or Hive team. Down to our last two presentations here. Our second to last here is going to be AI for neurofibromatosis. We'll go ahead and get started here in a moment.
Yeah, Lucas, just one thing about the audio that was breaking down. I think key is on the author of the previous presentation. If they're online, just make sure that your video file is not corrupted, and if there is any issue, please resend it to us so that we make sure in the final output of this presentation will replace with the correct audio. And sorry about the issues, and solve the issues as well. Thank you.
Awesome, and we'll get going with AI for neurofibromatosis.
Welcome to the presentation. This is Artificial Intelligence for Neurofibromatosis for NF, part of Hack for NF 2020, and myself, Schwan. So there are two approaches in this project. The first one is using artificial intelligence, specifically deep learning techniques, to automate the identification of plexiform and schwannomatosis tumors. So we distinguish between these two MRI images of these two tumors using deep learning. So first, a bit of background about schwannomatosis, which is also known as NF3 and is often characterized as an overlooked NF because there aren't a lot of cases surrounding this schwannomatosis. It occurs in patients with NF2 as well, and the phenotypes of this disease and NF2 are very similar. But geneticists and pathologists have recognized that schwannomatosis is distinct from NF2 and the other type of NF1 as well. So therefore, imaging plays a very critical role in diagnosing this disease, but it continues to remain unfamiliar to several radiologists as well. And it is imperative that radiologists be able to differentiate schwannomatosis from NF2 and NF1 on MRI imaging as well, because there are significant differences in the management of these diseases, the clinical outcomes for the patients as well. And then because of the significant differences in disease management and the patient mortality, distinguishing between between schwannomatosis and NF is more than an academic exercise on part of the diagnostic radiologist, and is very imperative as well. And typically the diagnosis of schwannomatosis is carried out using MRI technology because of its unparalleled capability in the evolution of such vertebral masses and in this field as well. And here's a bit brief about plexiform neurofibromatosis as well. And I obtained a data set from Synapse, this imaging data set, and also from Radiopedia, thanks to the doctors who put the radio images out there. So I built a CNN, which is a convolutional neural network for this particular task. This is a deep learning approach. I've shared the architecture. There are several layers such as the conv 2D layer, the max pooling, the dropout, then this dense, and I also use the ReLU function as well in this particular CNN. So CNN are basically, it replicates the design and connectivity pattern of our brain here. And achieved a staggering accuracy of 99.74% after 10 epochs. And there were 50 steps per epoch as well. So this is a very good sign. And these are the experimental results for the train and the testing data set. The data set was very small in size. Therefore, I also use data augmentation techniques such as zoom and shear scaling, rotation, zooming, and here. And these are the references that I use. So thus we use two very important artificial intelligence techniques. One was deep learning for distinguishing between schwannomatosis and the other NF tumors, because schwannomatosis is something which occurs very rarely. You don't have a lot of data set around it. There aren't a lot of expert radiologists who can easily identify whether a person is suffering from schwannomatosis or not. Therefore, we seek to automate this task and make computers work on our behalf for this. Because they will be more accurate as well, since you obtained a very, very good accuracy rate of 99.74%. The second.
Awesome. Thank you to the AI for NF team. I know he cut his recording there a little bit short in order to keep it within the time frame. And with that being said, we are down to our last presentation, which is the NF Care app. Let me get it pulled up here momentarily. And I know their project leader is in the chat. So if you have any questions for him, that's the last time I'll remind you, and we'll get started with our last presentation. Unfortunately, it looks like this is a corrupted audio file. So as Salvo just said to the previous team as well, if you could resend me your recording, we will be sure to replace that. But unfortunately, it doesn't look like the audio is coming through. But that being said, that is all of our presentations. So I just want to say one more big thank you to everyone that sent in their pre-recordings. I will go ahead and stop sharing my screen and pass it back over to Salvo.
Thank you, Lucas. These are amazing. Thank you so much, guys. I am so excited about seeing the results of this hard work that you did over the 6+ weeks that we launched this event. This is beyond our expectation, really. There are amazing apps and I think very interesting angles where you took research and data to find something new. I'm very, very impressed. I'm sure this is going to catch the attention of all our experts. We hope you'll stay engaged, because we want to continue these events in the future and further develop your ideas or new ideas and enrich our data sets that we can make available. I'm sure the community will contribute even more, seeing the excitement and the results and the depth of professionalism and mature approach that you actually apply to NF. We are very, very grateful for all your time. And we will make this video available for your sharing in a few days. Hopefully it will get fully captioned and you will be able to review all of the projects. As I mentioned, maybe not all projects that are submitted presented today, This was not a requirement for submission and competition. It was a way to showcase your project, because then we can use your 3 minutes pitch to advertise your project in the future. So thank you so much. You still have 12 hours or so and counting for your final submissions. If you want to apply for a grant prize, please submit that 3 months plan that we can review it. Thank you so much.